stationary phase and other is the mobile phase. There are two phases in chromatography. One is stationary phase and the other is mobile phase. And later on, depending on the partition means distribution of the mobile phase or solute and solvent, what you call, into the stationary phase. That is partition. Partition means distribution in the stationary phase. And based on that, different substances, they are separated. Now, like we can say, what is the partitioning? Like this is absorption and this is adsorption. If you are eating it normally, it is absorption directly going into your stomach. And if you somehow old Indian movie style, then it is adsorption. So in chromatography, I told you there are two main phases. One is the stationary phase and the other is the mobile phase. And chromatography, depending on this mobile phase, we can divide it into two techniques. That is liquid chromatography and gas chromatography. If the mobile phase is a liquid, we call it liquid chromatography. And if mobile phase is a gas, we call it gas chromatography. So depending upon this, we can characterize chromatography into three techniques: gas chromatography, gel chromatography, and liquid chromatography. And depending on the mobile phase and stationary phase, it can be gas solid, gas liquid, and gel chromatography can be filtration, permeation, and liquid chromatography can be ion exclusion, ion chromatography, partition chromatography, it can be thin layer or HPLC, and ion interaction, ion exchange, adsorption chromatography can be paper chromatography or reverse phase chromatography. Now, there is a very because in 1 hour it is difficult to control, difficult to give introduction of this technique. This is the sum of the backside of the, this is the diagram or photograph of the HPLC system. And that is how it looks. And this is another diagram, and I will explain you different parts. Like this is the injector, and these are the mobile phase. Here is the column and detector and data analyzer. So before these are the some of the diagrams of the instruments. Now, I will not go into details of all the parameters of chromatography, but some main parameter is like selectivity and resolution. Selectivity means our detector system is selective, means out of two, it can select one analyte. Out of two analytes, that is, it can detect one. Or we have developed a system that is a mobile phase and stationary phase in such a way that we can achieve selectivity. Either one of the component is removed or retained on the column, and later on we can remove it. And resolution means the separation between the two signals of the analytes. That is, we can separate the signal of the two analytes in such a way that we can do the analysis. So the retention time is given by TR, that is, it is the distance from the start to the main peak. And this is known as, in HPLC system, we get a small peak when the solvent system enters into the system, and we call it solvent peak. or this is known as T naught. And this width of the peak, this is known as peak width, and the overall height. Pointer see अपना. This is known as the peak width, means the total peak width, and the total area under the peak is directly proportional to concentration of the analyte. So from the peak area or from the peak height, these two things, they are directly proportional to the concentration of the analyte. And depending on that, we can analyze an analyte. Another some basic parameters like dead volume. Dead volume is the liquid volume, total volume of the liquid phase in the chromatographic column. And capacity factor, it is the ratio of the reduced retention volume to the dead volume. Means VR is the reduced retention volume and V naught is the dead volume. So this gives you the capacity factor. In chromatography, depending upon phase, stationary phase, we can divide it into two types. That is normal phase chromatography and reverse phase chromatography. In normal phase, if the mobile phase is non-polar and stationary phase is polar, then we call it as the normal phase chromatography. And if mobile phase is polar and stationary phase is non-polar, then we call it as the reverse phase chromatography. So like this, that is still depending upon the chain length of the silica, we can divide it into, we can characterize into mobile, we can reverse phase and normal phase chromatography. Now, in liquid chromatography, because we are applying high pressure, and when we apply high pressure in liquid chromatography, that is normally it is called HPLC, high pressure chromatography, or in another time it was called as high priced chromatography, because cost of the equipment was very high. And so in order to remove the bubbles present in the solvents, liquid nitrogen or by sonication, Okay. Now, from using another method for removing the bubbles is means the dissolved oxygen or dissolved air in the solvents is ultrasonication. We sonicate the solvents or the mobile phase for about half an hour, and all the bubbles or air which is dissolved in the solvents, it is removed. And after that, we can use the mobile phase. So we have to saturate either with nitrogen, or we can carry out the we can carry out the removal of dissolved air by ultrasonication. So this is like when we inject, when we move, when we allow the mobile phase to pass through it, solvents have a little bit absorption in the detector, and we get a solvent front, and after that it becomes normal. So this is a solvent front. दो खोली हुई है।
बस स्टॉप प्रेजेंटिंग कर दिया नहीं, उसको स्क्रीन नहीं आ रहा, यूट्यूब में नहीं आ रहा। अच्छा, मैं तो उन्होंने नहीं किया द टैब स्क्रीन मैं कर लूँगा मेरे बाद।
क्या हुआ?
HPLC
अच्छा, so I told you we can degas the mobile phase either by sonication or by applying partial vacuum or by passing helium gas, or in some instruments there is a online degasser system that carries out the uh, degasification. Now, gradient in chromatography, in a liquid chromatography, there are two systems. That is, one is gradient system, and other is non-gradient system. In gradient system, we can pass the mobile system of different polarities for a fixed time. And in non-gradient system, we can run a single mobile phase. In gradient, we can fix the time of the one solvent for a particular, that is, we can run the mobile phase for a particular time. Like if we want to run a mixture of acetonitrile water and methanol water, or any other solvent based on polarity, we can have a acid methanol, acetonitrile, or ethyl acetate as the different mobile phase at different times. So we can carry out the separation in the gradient mode and in non-gradient mode. So as I told you, peak width, and peak width gives you uh, the base. It is calculated in a Gaussian way, and this is the area under the peak width, peak of the analyte. And the half of peak is known as the half peak height. That is, suppose you have uh, from here to here. its half is peak height. And we can calculate the ratio, the standard deviation and the other things. Another thing, sometimes what happens, the injector of the column is clogged. Like, and this we can know, like if we get in the first run, we get a bigger peak, then the size of the peak key is keeping on decreasing. And this happens when our injector is clogged. Clogged means there are some impurities of the analyte or some impurities of the solvent, or we have not done the proper filtration and sonication of the uh, mobile phase. Then this injector gets clogged, and it is the mobile phase, and the sample is not able to pass into the detector. Now, how we can maintain the pump? In HPLC, there is a pump, and it is very important to take its care. And for maintaining the pump, we must wash the pump with a buffer. And there are seals in the buffer, and those should be replaced after a regular interval of time. And we should not allow any solids in the mobile phase. And another thing is, from the reverse phase, the mobile phase, reversed mobile phase, if there are any buffers, we must wash the column regularly after uh, different ratios of water, so that all the buffers are removed, because buffers, they contain the salts, and salts, after the evaporation of the solvent, they get precipitated out. So in order to avoid the precipitation or clogging, these salts, they must be removed with water, depending on the uh, so how HPLC is useful in pharmaceutical industry? No doubt, for HPLC, liquid chromatography is very useful in environmental, in analysis, and also in pharmaceutical industry. It is very, very important. Like it is used in manufacturing, content manufacturing process, like it is used for content uniformity. Like if you have a drug, and one part of the drug is having more content, or one tablet is having high concentration and the other tablet is having low concentration, then in order to check the uniformity, means all the tablets, they are having the same concentration. Like paracetamol, if you have different concentrations of paracetamol in different tablets, then it is a for the patient. And dissolution and stability studies, that is for all these purposes, it is used in manufacturing. And in the discovery, like purity and concentration, redox potential, sometimes we can confirm the structure and metabolite and half-life in the body. Like this is how we can see the content uniformity of a drug. Like if even there is little bit impurity. Now you must see that in pharmaceutical uh, industry, it is very, when we manufacture the drug, some of the solvents in which drugs are manufactured, they are left in the drugs, and these solvents are highly carcinogenic or poisonous for the human body. So it is necessary to know these solvents and their concentration in the drug samples. So sometimes another, that is, a drug is decomposed. Like this is your normal chromatogram. If the drug is decomposed, then you will get another peak in your chromatogram. And this indicates that somewhere your sample is not pure and it has some contamination, and this contamination is indicated by this impurity which is detected in the detector. So these are different applications, and so I will come. There is a competition between HPLC and gas chromatography. In HPLC or liquid chromatography, we do not require any volatile substance. And the samples, they must be soluble in the mobile phase. And in gas chromatography, the necessary condition is that the sample, it must be volatile. And in gas non-volatile, in gas chromatography, the sample should be volatile, and in gas chromatography samples, it can be non-polar or polar. And by this way, we can have, depending on the polarity, and that is a hydrophilic or hydrophobic character, or volatile or non-volatile character, we can distinguish between the different analytes. Now, how the HPLC works? I told you there is a solvent, and from this solvent there is a pump, and there is a column detector, and detector is connected to the data evaluation system, that is a computer or software that analyzes the data. And this separation, this can be done in the column, and the column is a stationary phase. It can be a C18 or silica, and depending on that and how the separation takes place in HPLC is like this, that sample is injected and we get the chromatogram. Then depending upon this, like we can analyze it. Like this is T naught, this is the, I told you this is the solvent front. This is TR retention time for one component, and this is the retention time for the second component. And depending upon the standards, you can analyze your substance. So depending on this, how a modern this HPLC looks like mobile phase and injection column, it can be controlled by a temperature and the wavelength. So depending upon this, we can apply HPLC in different areas, like for chemical, bioscience, consumer products, clinical, environment, and pharmaceuticals. This is little bit introduction of HPLC. And in our lab, our students, they have done a lot of work on HPLC for the separation of various analytes, either pharmaceuticals or environmental samples or cosmetics, consumer products, all type of samples, they have been analyzed in our lab, and day and night we have about HPLC system with UV detector, uh, fluorescence detector, and uh, photodiode array detector. So different analytes we have analyzed, like fluoroquinolones. I will not go into details, but because time is short, fluoroquinolones are the antibiotics. And you can see you can see the structure of these fluoroquinolones, and these fluoroquinolones, they have almost similar structure, and it is very difficult to separate these. But we have, this is like I told you, this is the solvent front, and all these fluoroquinolones, they were analyzed. Like ofloxacin, this ofloxacin is used as a eye medicine in your eyes. Nalidixic acid is used for your stomach pain, and all these fluoroquinolones, that is, they are used as antibiotics. Similarly, we have applied gradient HPLC, and then you can see these fluoroquinolones, like which were here, not so well resolved, not so well separated. They get very well separated, and you can uh, analyze them very uh, easily. So these are the analytical parameters, and if anyone needs, we can provide them. And similarly, we have applied this in urine samples, in water samples, in hospital waste water, and in pharmaceutical samples, and nalidixic acid, or this is used for the ट्रीटमेंट ऑफ चिकन्स. जो चिकन खाते हो ना आप, उसमें बहुत अमाउंट इन फ्लोरोक्विनोलोन्स का, नैलेडिक्सिक एसिड का अमाउंट ऑफ नैलेडिक्सिक एसिड इन chicken, and different parameters. Similarly, we have analyzed aldehydes. You see aldehydes, they are like you have almonds. In almonds, aldehydes are present, and like cinnamaldehyde is present, and we can detect these aldehydes in like cinnamaldehyde we have found in almonds, and benzaldehyde we have found. So these can be these analytes. This is the simple little bit of the small amount of the work I shown you, that we have analyzed liquid chromatography, and mass spectrometry is another world. Both techniques are different. It is very difficult to combine liquid chromatography with mass spectrometry. Mass spectrometry is a uh, uh, very good detection, online detection system for liquid chromatography. The discovery of mass spectrometry started with in 1897 from the J.J. Thomson's experiment, and after that in 1983 till then a thermospray, this was developed. And mass spectrometry, it is based on the technique, analytical method to measure the molecules or atomic weight of samples. And in liquid chromatography, mass spectrometry, what we need to combine, like we start with mobile phase, it enters into column, then ionization and mass analyzer, that is in the column, separation takes place and the ion source detects the ions, and we can handle the data acquisition with the data acquisition system. Mass analyzer is under a very, it has a low pressure. That is, now mass spectrometry, as I told you, this is J.J. Thomson's, Sir J.J. Thomson and our TV or oscilloscope and Dempster's experiment, they all projected the mass spectrometry. So mass spectrometry, you can see that we can detect all the elements of the periodic table. We can molecular weight, we can find the atomic weight or molecular weight of a substance. Nowadays, mass spectrometry is highly advanced technique for analyzing the systems. And basically, the mass system and the mass spectrometer consists all like it has an inlet from where we inject the sample. Either it may be coming from HPLC or gas chromatography or any other analytical technique. Then it enters into the ion source, and depending upon ion source, mass spectrometry can be matrix assisted laser desorption ionization, electrospray ionization, ion spray, fast atom bombardment, liquid spray ionization, mobility mass spectrometry, electroionization, or chemical ionization. That is, this depending on the ion source, different types of mass spectrometry they are developed. Then the ions are produced, then they enters into the mass filter and carried to the detector and then to the data system. In the data system, evaluation is done using a mass, using a suitable software. So main thing I show you how the mass spectrometer works. Like we inject the sample. Let me tell you first the different components. This is the ion source. This is under high vacuum. Then the ions which are produced, they are passed through a electric and magnetic field. And from vacuum pump, we apply a high vacuum inside this tube. Now, depending on their mobility, depending on the mobility of the ion which are produced, I will show you what is the basic principle. When the electrons, they are produced and they bombard the sample, and by this way ionization is produced. Like you have a atom molecule, you bombard it with an electron, then this molecular ion is produced. And depending on the mobility of this ion in the vacuum tube, different ions, they get separated. Depending on the mobility, that is half mv is equal to zv, we have allow the ions to move into the electric field and magnetic field, and depending on their mass by charge ratio, these ions get separated. So how to predict the mass spectrum? Like this is the mass spectrum which we obtain, and we get different peaks. And what can happen when the electrons which are produced by the ion source, they bombard with the atom or molecule of the analyte, it it gets decomposed, and different fragments are produced. Depending on the fragments, they are detected by the detector, and based on the fragments, we get different peaks in our mass spectra. First is the molecular ion peak, and we call it, and other is base peak, and we call fragment ions. These are the fragments which are produced. And base peak corresponding to the main molecular ion which is produced in the system. I will show you some, and fragment ions and some of the uh, ions, they may not be stable, and there is metastable ions are produced, and these metastable ions, they try to uh, convert themselves into a stable ions or neutral fragments. And depending upon these fragments, uh, we can have different ions or characteristic molecular ion peaks are produced, and they help in the identification of the analytes. In our SICI, the Sophisticated Instrumentation Center, you should visit. There is a liquid chromatography LC top MS system. And from that, you can, in that system, like you bombard in mass chromatography, basic principle is you inject the molecular ions. You inject the molecules. Either they are coming from gas chromatography, mass, gas or mass, it doesn't matter. Then they come bombard, they are bombarded with the electrons. Ions are produced in the vacuum tube. Molecular ions are produced, and this may undergo fragmentation. The molecular ions which are produced depending on their movement in the vacuum, they get different fragments which are produced. You can capture those fragments in time of flight mass spectrometer. That is, you can work on the different fragments. That is, this fragment is from this molecule or another molecule. That is, you can undergo very deep into the molecular characterization. So there is a like ethyl bromide, and depending on its molecular mass, m by z values, you can see if it is producing a m positive ion or m2 positive ion, that is produced, and depending on their abundance, you can see the concentration values. So these two techniques, they are combined together, and that is, and these are known as liquid chromatography, mass spectrometry. And Emicizumab is a monoclonal antibody, and this is used for the treatment of prophylaxis in several hemophilic patients. So this is the journal reference. If you like to see, you can consult it. And like if we see a blank, this comes out in this. When we inject the drug sample, it this peak at a retention time of 1.6 is produced, and depending on this, that small amount and higher amount, you can make a calibration curve. So another application is in LCMS is in the analysis of homoserine lactones, and this gas chromatographic work is done in our lab, and later on LCMS work is reported by some other workers, and this work is presented in Talenta. Now, like homoserine lactones, these are produced by the Pseudomonas aeruginosa bacteria. Homoserine lactones and the bacteria, they communicate with each other through secretion of these homoserine lactones. ਜਿਵੇਂ ਆਪਾਂ ਕਹਿੰਦੇ ਨਾ ਕੀੜੀਆਂ ਚੀਟੀਆਂ ਮੀਟ ਇਹ ਵੀ ਗੱਲਾਂ ਕਰਦੀਆਂ, ਹਾਊ ਦੇ ਟਾਕ। ਔਰ ਜਿਵੇਂ ਤੁਸੀਂ ਦੇਖਿਆ ਹੋਣਾ ਇੱਕ ਬਟਰਫਲਾਈ ਹੈ, ਇੱਕ ਆਉਂਦੀ ਹੈ ਉਹਦੇ ਪਿੱਛੇ ਪਿੱਛੇ ਦੂਸਰੀ ਆ ਜਾਂਦੀ ਹੈ, ਇਸੇ ਤਰ੍ਹਾਂ ਹਨੀ ਵੀ ਇੱਕ ਆਉਂਦੀ ਉਹਦੇ ਪਿੱਛੇ ਪਿੱਛੇ ਸਾਰੀ ਆ ਜਾਂਦੀਆਂ। ਹਾਊ ਡੂ ਦੇ ਟਾਕ ਵਿਦ ਈਚ ਅਦਰ? They talk with each other through the secretion of some hormones, or you can see these bacteria, they talk with each other through N-acyl homoserine lactones, and in nature they produce different, depending on this chain length, that is 2 carbon, 3 carbon, 4 carbon, 5 carbon, 6 carbon, 7 carbon. And not only this, they are present in nature as stereoisomers. There is dextrorotatory and levorotatory. And another interesting aspect is that one isomer is active in talking and the other isomer is not active. So depending on this, that is these homoserine lactones up to 10 carbon atoms, they and their isomers like C4, dextrorotatory, levorotatory, C6, dextrorotatory, levorotatory, their isomers can be separated using the columns, and we can detect the basic peak. If in your substance it appears at 57 mass m by z peak, then you can see that you can confirm that homoserine lactones are present in your sample. And these homoserine lactones, they are also responsible for cancer in the lungs. So by this way, we can detect and apply LCMS substances. Thank you very much. And if you have any question, you can ask. We have 5 minutes, and after that you will get your tea. Hanji, ਕਿਸੇ ਨੂੰ ਵੀ, if anyone has any question. And we have a break of 1 hour. At 12, you come to Dr. Olak. He will, on the first floor, you will be introduced to HPLC working, right? And you can see how this system works. And this was the introduction to liquid chromatography. And we will try you to bring to the MS system also. We have a small MS system in our department, but in instrumentation center, there is time of flight mass spectrometer. This was the brief introduction to LCMS system.